To the last show here. Really, what we're looking at now is more of the physiology about how does the eyeball go about actually taking this light information and doing something with it, along with how do we actually focus it in on the retina. So light again is refracted, and again refraction is when light passes through substances of different densities. When it's entering and leaving those different substances, it does tend to bend the light rays. If you've ever taken like a fishing pole or a fishing net and put it underwater, it looks like there's a bend in it. And again, that's really just from the refraction of the light leaving the water and going into the air. So we bend this light a number of different times when it's going through the cornea, when it enters the lens, and when it leaves the lens. And it's really the combination of this refraction of light doing all these things, along with the fact that we can change the shape of our lens, as well as control light intensity with the pupil, that we can actually focus this image onto the retina and get some type of signal. Normal vision is when everything's focused normally. There's hyperopia or farsightedness. In reality, what's going on here is if this was the back of the eyeball right here, in hyperopia, the image is actually coming into focus here. So when it's hitting the retina right here, it's actually out of focus. You can fix that with lenses. Myopia is nearsightedness. The eyeball in this case is too long, so instead of the signal being focused right on the retina, it's actually focusing at a point in front of the retina. So by the time the light's hitting here, it's out of focus. Again, corrective lenses can fix this. Astigmatism is really one of those refractive surfaces, or the back of the eye might have an abnormal area. It just means that it might be focused in one spot, but one part of that image is not in focus. So a lot of times it's an irregularity on the lens, on the cornea. Again, we can do this by having either weighted contacts, where they always reorient, or by doing glasses that have certain areas that are slightly different than the rest of the lens. Generally, as you get older, the lens has harder and harder time going back to spherical, which means as you get older, you start holding stuff further and further away to read it. So as we look at what's going on with these actual receptors, this is where things start to get a little bit more interesting. So in terms of the number of photoreceptors we have per retina rods, you have about 130 million. So combine that's 260 million between the two eyes, over a 250 million receptors. These really are detecting presence or absence of light. These work really well in night vision, detecting movement, but are not doing color vision. What is going on here is these have a particular pigment in them called rhodopsin, which one of the things that helps derive this is vitamin A, which is why they say vitamin A is good for your eyes. But when light hits this rhodopsin, it is actually going to cause a change in this molecule. The other thing we'll see on this one is it takes quite a while for this rhodopsin to be regenerated, which is part of the reason if you're in a bright light area where this photopigment is going to be pretty much bleached out in this bright light area, and then you go in a really dark room at a theater when theaters used to be open and we weren't in a pandemic, and you go into that theater, all of a sudden it's really dark in there, and it takes a little while for your eyes to adjust. That is that rhodopsin being regenerated so you can actually see in this lower light environment. But again, these rods get triggered is when light hits that photopigment. What is interesting about the eyes is they don't work a lot like everything else we've talked about. Generally, something is not active or it's not depolarized, and we're actually bringing about a change that leads it to getting to threshold, depolarizing, and bringing about a signal when we've talked about the nerve stuff here. Eyes are a little bit different in that these photoreceptors in darkness are normally depolarized. They are generally sending a signal to the upstream cell all the time. This sodium is generally always entering into the cell, and we have what we have going on here called dark current. This causes them to send a neurotransmitter up to the bipolar cells. What is interesting on these ones is with this dark current, it's always an inhibitory neurotransmitter to those bipolar cells, which hyperpolarizes those bipolar cells. So the upstream bipolar cells are not sending a signal when it's dark, because these cells, the photoreceptors, are always sending a signal saying, don't depolarize, we're going to keep you hyperpolarized. They don't stimulate a ganglion cell, no nerve impulse is generated back to the brain. When light hits the photoreceptor cell, it is going to lead to a hyperpolarization of that cell. And what this does is it causes reduced sodium entry into these cells, which means these cells start to hyperpolarize. As these cells start to hyperpolarize, they stop having this dark current and they stop releasing, they block those calcium channels like I was saying, and they stop releasing this inhibitory neurotransmitter. So these bipolar cells now are no longer getting an inhibitory signal. They can generate their own graded potential, and they can send neurotransmitters to the ganglion cells, which can reach threshold and send a signal to the brain. So to stimulate with the eyes, the photoreceptors, what you're doing is you're inhibiting the inhibitor to stimulate, which again is kind of a backwards way to do it, but this is how the eyes work. You inhibit the photoreceptor cell, which is normally inhibiting the bipolar cells from sending a signal. Once you inhibit those photoreceptor cells, the bipolar cells can then send a signal to the ganglion cells, which can lead to a nerve impulse going back to the brain and to you seeing something. So again, we're inhibiting an inhibitor. It's like subtracting a negative to get a positive. It's kind of different. But I always think that one's really interesting and kind of confusing to people, in that you inhibit an inhibitor to stimulate instead of just stimulating. Why that's the case, I don't know exactly why that's the way it evolved, but this is how our eyes work. So this is what a rod looks like. And again, this rhodopsin is that very light sensitive one, very, very sensitive. One photon hitting is enough to bring about a change in it. It takes quite a while for these pigments to get fully recharged, so it really takes about 30 minutes of dark to get to a fully dark adapted state. Like it says, there's a single rod will hyperpolarize if there's a single photon of light that hits it. In bright lights, we bleach these out almost completely, which again is why if you go from a really light area to a really dark area, it's very hard to see, because it's really our rods that are doing low light vision. And when they're all bleached out, you can't get a good signal at that point. Because vitamin A is key in making rhodopsin, if you have a complete lack of vitamin C, you will not be able to see well at night, because you're not making enough sufficient rhodopsin to have a good sensitivity to these states. Cones are a little bit different, not nearly as many of them. They're kind of intermixed, but generally in our areas of sharpest vision. These are going to have actually multiple different types of pigments. So they have what's called idopsin, but there's 3 different kind of chemicals within that. You have the red, green, and blue cones, which have slightly different forms of idopsin. And it's these that, based on the wavelengths of lights that are hitting them, can respond slightly differently, which will let us perceive colors. So we only have about 6.5 million of these per retina, mainly in the areas of sharpest vision. Relative to the other ones, it takes a lot less time for this to be regenerated, which is part of the reason we use these in higher light conditions. And again, these certain types of cones have slightly different absorption spectra, which means the red cones that we're going to absorb light best in the red spectrum, green cones are going to be more in the green spectrum, and then the blue ones in that spectrum. And again, it's kind of the percentages or how much each of these is getting stimulated, which is going to let us perceive these different colors. If you have a red cone that's getting stimulated a certain amount, and a blue cone that's getting stimulated a certain amount, and then the green not being stimulated at all, you might be getting something, okay, that says this is a purple based on these ones, this percentages that are being activated. So again, that color that's perceived depends on how much stimulation there is on each of these different types of cones. So you can see the rods are much smaller than the cones. So these are the rods, these are the cones. And again, different spectra here. The rods, they are kind of in the middle of this one here. This is their max absorption area at about 500 nanometers. You can see blue cones absorb light best at 420, green cones at 531, red cones at 558. Again, not that I'm going to ask you these numbers, but their absorption spectra are a little bit different. So when you hit it with a particular color of light, so like blue light would stimulate this one and part of the rods, but might not stimulate the green cones and the red cones at all. where red light is not going to stimulate that blue cone or very much of the green or the rods at that particular wavelength. And this is what I was kind of saying, depending on how activated these certain ones are. Orange light, it might be that you're getting the good percentage of the green cones and almost all the red cones. We perceive that as orange light, where blue light, you're stimulating only the blue cones and not the red or green cones. So we perceive those ones being activated as that what we're seeing as being blue. And again, white light is a combination of all these ones, so they're all being stimulated equally if you're seeing white. So again, how this is working, light is entering the eye, being refracted by the cornea and the lens, well, by the cornea through the pupil into the lens. The lens is going to further refract this and help focus this light onto the retina. We can change the shape of it depending on if we're far away or closer. These photoreceptors, when they're hit with this light, are hyperpolarized in the presence of light. That stops their inhibition of a bipolar cell, which lets the bipolar cells release neurotransmitter. That allows those ganglion cells to be activated and send an action potential through the optic nerve back across what's the optic chiasm, where these cross. Not a big deal in this one right now. That is going to go back to the brain, through the thalamus to the visual cortex, and then we determine what this actually means, how we perceive this. There is some differences in people's color vision. So there is, it's on the X chromosome for some of these red and green cones. So red-green color blindness is a sex-linked trait, is what they refer to it as. It's found in the X chromosome, much more common in men in that we only get one copy of the X chromosome. So about 8% of males, of which I'm one of them, has some red-green deficiencies. Again, you don't see red and greens as being entirely distinct from one another. If you've ever looked at those little Ishikara plates where they have the different little dots with the numbers in them, I can't see a lot of those ones. But that is that absence of having a certain amount of cones here. Again, when we start talking about the fovea centralis, why our straight-on vision is so good is that we have very little convergence happening. So you might have 4,000 cones going to 4,000 bipolar cells, but when you're out in your peripheral vision, it might be 600 rods going to one bipolar cell. So the eye does have a lot of convergence. In all honesty, there's a lot of processing that takes place in the eye. Again, there is no photoreceptors on that blind spot, but our brain does kind of take the peripheral areas around that and kind of fill that in. So when you have one eye open, let's say, and you're looking outside, you don't see a black spot where your optic nerve leaves your eyeball. You don't see a blind spot in there. And it's mainly because your brain kind of takes that and fills it in based on what's surrounding that area. What this really is saying, this is really it for A&P1. So this is the last chapter here. We'll be getting, over the next couple weeks, having a chance for you to go through all these videos. Final, again, is going to be over nervous system and senses. Main thing is it's been great teaching all of you this semester. I know it's been a weird year, 2020. What can you say about it? Really nothing much good. But sorry we've had to do it this way. We're going to have AP and 2 in the spring being a little bit like this, but hopefully in the next year or so here, college will start going back to a little bit more normal. But it's been a different process, but it's been a joy to teach all of you, and I hope this has worked out okay for you.